but 108 women this time. We again collected the microbiome on the forehead and performed qualitative PCR for Corynebacterium caseri, quantify its presence, and also we detected the presence of Cutibacterium acnes, that is known to decrease with aging, and Corynebacterium propensity, that is known to increase with aging. Regarding the clinical parameters, we perform instrumental measurements on the skin barrier, hydration of sebum levels, but also mechanical properties of the skin, and clinical scoring based on images of the face, on wrinkles, pigmentation, and complexion. I have to say that the thermometry and chroniometry values were not significantly different between the two age groups, so I'm not going to talk about these parameters later. So when we got this data of the amount of bacteria and instrumental measurement, we performed statistical analysis with Spearman correlation coefficient, and I'm going to show you this correlation. So, indeed, the Corynebacterium caseri decreased with aging, but not only. So here you have the representation on this graphic of the Spearman correlation coefficients. The green bars reflect positive correlation between the bacteria, the amount of bacteria, and the clinical parameter. Red bars, the negative correlation, and the gray are not significant associations. So as I said, the amount of C. caseri decreased significantly in the old group, but also was associated with, decreased with the presence of pigmented spots and wrinkles, but on the contrary was highly associated with young skin parameters like more radiant skin and more elastic and firmer skin. So we performed this association for C. acnes and propensity also. And we confirmed for C. acnes that this bacteria was highly associated with high level of sebum and was associated quite with all the parameters of the young skin we analyzed significantly. And on the contrary, Corynebacterium propensity was behaved the opposite way. It was associated with the old group and the presence of wrinkles and less firmer skin. So we can say that the evolution of Corynebacterium caseri evolves and resembles more the evolution of C. acnes. So, but at this stage, we just got a significant association. We don't know if the decrease of C. caseri is just a consequence of aging or if this bacteria can have benefits for the skin. So to answer this question, we co-cultivate the bacteria with on the top of 3D skin model. Skin model. And so we use to do so, we use two different concentrations of the bacteria, and we manipulated on the top of the skin and compared it to the control model with the medium alone. So after 2 to 5 days in culture, we analyzed the bacterial growth first, then the regulation of genes, the skin genes by transcriptomics, metabolite regulations, and also, because we like to see the bacteria on the skin, we perform scanning electron microscopy just to look at the bacteria at the end of the culture. Regarding the bacterial growth, after 2 to 5 days, we just scrubbed the surface of the skin and collected the bacteria and divided the sample in two. One sample to count the bacteria, the ones that are able to grow on agar plates, and the other sample for molecular biology. Qualitative PCR can show all the bacteria, living or dead, and PMIQ PCR focuses on the living bacteria, those that have intact membrane. And so what we observe regarding the growth of Corynebacterium caseri is that between day 2, when we inoculate, day 0, and day 2, indeed we observe growth of the bacteria on the skin and using the 3 methods. After 5 days, the number of bacteria on the top of the skin stabilizes the total amount of bacteria. And when we look at the number of bacteria that are able to regrow on agar plates, this number has slightly decreased. But when you look at the bacteria using scanning electron microscopy after 5 days in culture, we are still able to see bacteria living in quite a high amount on the top of the skin. So we know the bacteria is able to grow. Then what happens at the level of the skin response? We perform DNA array by extracting the whole skin model and looking for gene regulation in the skin using DNA array, sorry. And when we compare the number of genes regulated by the low or the high concentration of bacteria, we found that some genes were regulated in common by these two conditions, but the highest concentration of Corynebacterium casiri induced the highest number of gene regulation, more than 1,000 genes. And I will show you an example of pathways. The pathways related to keratinocyte proliferation and differentiation were the most regulated. And here you have example of genes that are related to differentiation that are decreased, such as some keratins, loricrin, corneodesmosine, and on the other side, upregulated genes related to proliferation. So the presence of, this suggests that the presence of Corynebacterium casiri maintains the skin model in a more proliferative state. And we confirm this result at the metabolic level, looking at the whole skin model metabolites. So in this case, we still have the bacteria on the top, so metabolites we observe could be produced by the skin, by the bacteria, or by both cells. We extracted it in different extraction types and analyzed by chromatography. The first thing that's quite interesting is that the main driver of metabolic change in the model during the culture actually is time. When you look at the difference between day 2 and day 5, they are really well separated, these conditions, when we represent the data in this principal component analysis. So that's quite logical because, as you know, the 3D skin model is a living model, so it's still evolving during the culture period. And here is the control model. You see that the epidermis is thinning, the living part is thinning during the different the culture, while the stratum corneum is thickening. So it's quite logical to observe strong metabolic changes. But when you look at specific time points, you can see that data are not well separated at day 2, not much is visible, but at day 5, the control and the bacteria colonized model will separate again. And indeed, there are more metabolites that are significantly up or down regulated at day 5 compared to the control. And we did look at those metabolites, and the most regulated pathway we observed was the superlipid pathway. Number of metabolites, lipid metabolites were downregulated and compared to the control condition. But if we look into detail, for example, this ceramide here, you can see that it increases with time in culture in the control model, which is logical with what we've seen before, the differentiation process. In the control, in the model colonized by Corynebacterium casiri, there is not a decrease, but it's not increasing so much. So the difference is significant. So there is like a slowdown of the epidermal differentiation in the presence of bacteria. And on another side, there is some markers, some metabolites that could reflect a more proliferative state. For example, the most increased metabolite we observed was adenosine. Adenosine, you can see that in the control model is decreasing with differentiation. While in the model colonized with Corynebacterium casiri, it's slightly increasing. So there is a huge difference between the two conditions. And I haven't shown this data, but internally we did also bacterial culture, and in bacterial culture, Corynebacterium casiri was able to produce adenosine. So here in this model, we don't know if it's the adenosine produced by the bacteria, but in the whole model, we have more adenosine. And adenosine is already used as a cosmetic ingredient, anti-aging ingredient. We know that it can activate its receptor in the keratinocyte and participate to proliferation by increasing DNA and protein synthesis. Another marker was the example here is cysteine glutathione disulfide. That is also decreasing during the differentiation process in the control, while it's not decreasing so much in the bacteria colonized model. And we observe like an oxidative stress in the model. So saying like this is not really nice, but actually ROS, reactive oxygen species, are normal byproducts of mitochondrial metabolism. So we think that the fact it decreases less during time in the bacteria colonized model might reflect more proliferative and more metabolic activity in the cells. So to summarize, the tool we used, Culturamics, was really useful to discover new bacteria on the skin that could play a role on skin health and skin aging. So because we need to have those bacteria in the lab to study them. And in our case, Corynebacterium castori decreases with aging, associated with young skin parameters, but it's not just a statistical association. When we use these bacteria in contact with cells, we also observe the benefits maybe on skin renewal, epidermal renewal, maybe by producing adenosine. So of course it would be interesting, and that's work we are doing at the moment, to increase the level of Corynebacterium castori in the old skin with using prebiotics or even using postbiotics from this bacteria to benefit the skin. So I'd like to thank the team at L'Occitane in my lab, the skin biology, but also in the microbiology and clinical test teams, and also the team of Professor Bernard Escola at the Aix-Marseille University, who performed all the Culturamics studies. I thank you for your attention.